Hello everyone, this is the Theoretical Doctor and welcome back to my channel. It's been a long while since I've posted a video in the STPM Biology Semester 1 Chapter 1 series. Hence, for the subsequent videos that I'll be uploading will be on completing this series. If you want access to flashcards on this topic and other videos, do click the link below. It is available on my Pinterest and slides on this topic are available on my website. Links are in the description below. In this video, we will be continuing the subtopic of proteins and we will dive into the different level of protein structures whereby I will be focusing on the primary, secondary, tertiary, and quaternary structures of protein. Level of organization of protein structure. So there are 4 levels of protein structure to take note of. First, you have the primary structure, then you have the secondary structure, tertiary structure, and finally quaternary structure. Primary structure refers to a sequence of amino acids in the polypeptide chain linked together by peptide bonds in a specific linear sequence. Each primary structure is unique and determines the shape of the protein molecule and properties of a protein. So each primary structure is unique as they are controlled by the base sequence of gene, which also determines the function of a protein as it controls the other higher levels of structure. It is basically the foundation, hence which is why it determines the shape of the protein molecule and the properties of the protein. If there were to be any changes here in the primary structure itself, the overall protein molecule would be something totally different. Next, any changes, meaning any substitution or deletion in the primary structure can cause changes in the other levels as I mentioned before. For example, primary structural change of the 6 amino acid in the beta gene of hemoglobin from glutamic acid will change to valine, leading to sickle cell anemia. In sickle cell anemia, the shape of the cell is in crescent shape, like a moon, instead of the regular round or oval shape of the hemoglobin, or in other words, the red blood cell. The shape of the sickle cell is in crescent shape and has lost its elasticity. Moving on, the secondary structure. The secondary structure refers to alpha helix coiling and beta pleated sheet of the primary structure stabilized by hydrogen bonding. The primary structure was linear. That was what I talked about earlier. Here, the secondary structure of the protein is the localized repetitive coiling or folding of the polypeptide chain due to hydrogen bonds formed between the peptide linkages. Basically, you take a sheet of paper. The paper is the primary protein, okay? But if you were to keep twisting it, keep on twisting it, you'll get the alpha helix structure. But instead of twisting it, you were to keep folding it. Just keep folding it, like you know how we make those paper fan last time. So if you were to keep folding it, you will get the beta pleated structure, and pleated means folding. After you've twisted it or you have folded it, there are hydrogen bonds formed in between these. So the hydrogen bonds formed in between the twisting or folding maintains the stable structure of the alpha helix and the beta pleated sheet, which is why the structures are able to stay as it is. Okay, so alpha helix structure, it is coiled, extensible, and springy. For example, the keratins of fingernails, hair, wool, horns, and feather. In the keratins of hair, there are presence of disulfide linkages. This is just some extra information. And in the beta pleated structure, it forms from the polypeptides being folded, as I mentioned earlier, to form the beta pleated sheets. An example would be silk. It's composed of protein fibroin. Moving on is the tertiary structure. Tertiary structure refers to the folding and coiling of the already coiled primary structure of the polypeptide to form a globule shape. Globule is a 3D shape that is mainly stabilized by hydrogen bonds, ionic bonds, disulfide bonds, hydrophobic interaction, and van der Waal interactions. Not van der Waal, I made a mistake here, it's van der Waal. Each type of protein has a unique 3D structure, or in other words, they call it conformation, like conformation of protein, they're referring to the 3D structure, not ED, I made a mistake again, and has a specific function. For example, enzymes, antibodies, hormones, and myoglobin. Myoglobin is the oxygen storage red pigment found in muscles. It's similar to red blood cells, but it is only found in muscles. You'll learn more about this in semester 2. And finally, quaternary structure. Quaternary structure refers to a protein with more than one polypeptide, each with its own tertiary structure or subunits to form a single functional molecule. The structure is maintained by hydrogen bonds, ionic bonds, hydrophobic interaction, and van der Waal interactions. Van der Waal. An example would be hemoglobin and also immunoglobulins. Here, you combine multiple tertiary proteins to become one quaternary protein, and the overall structure is usually globular in shape. Remember that hemoglobin is a form of quaternary protein structure, whereas myoglobin is a form of tertiary protein structure. Do not get confused between these two. Alright, so here is just an overview of the levels of protein structures. Here's the primary structure, it's linear polypeptide chain, and then if you were to twist it, you get the alpha helix. If you were to fold it, you get the beta pleated sheet, and then after it has already been twisted and folded, and you twist it and fold it again, you get the tertiary structure. You have more bonds forming over here, and if you were to combine these tertiary structures and all, you will get the quaternary structure. Just a summary: primary structure, it is a sequence of amino acids. It determines the overall function of protein. Any deletion or substitution, anything that has been changed or damaged here, it will just change the whole protein. It's the foundation. Secondary, it consists of alpha helix or beta pleated sheet. Sometimes in exam they'll ask, what level of protein structure it is. It's an alpha helix, so the answer is secondary. It determines the regular coiling or folding in fibrous proteins. Tertiary, just remember 3D, that's where the 3D shape begins. Over here it's usually globule in shape. So it's 3D shape. It determines the function of globular proteins like enzymes, myoglobin. Quaternary structure consists of 2 or more subunits. It's when the proteins combine. It determines the 3D shape of some globular proteins, just like hemoglobin, red blood cells. If you've reached this point, thank you very much. Give this video a like if you found it helpful, as it does help in reaching out to others as well. To support my channel by pressing the subscribe button so I will know, and it gives me motivation to continue producing content like this in the future. And finally, share this video to those who might find this helpful. See you in the next one.